of CRISPR-Cas9 components. But before we dive into that, I just want to share why I find this so fascinating. So DNA sort of shapes who we are as individuals. It's the reason why I have black hair and you have blonde hair and you have red hair. But DNA does more than determine our eye color or hair color. It could be the reason why someone has sickle cell disease and someone else doesn't. Genetic diseases like sickle cell disease are caused by mutations in our DNA that disrupt normal biological functions. In fact, about 65% of people have some sort of health issue due to genetic diseases. So clearly this is a really big problem. Now, turns out we can actually edit our DNA using a technology called CRISPR. So CRISPR technology is made up of two main components, the Cas protein and the guide RNA. The guide RNA acts sort of like a GPS system. So it contains the same basis as the DNA that you're trying to target, and it guides the Cas protein to this strand of DNA. The Cas protein is kind of like a pair of scissors. So it creates a break in the DNA at this point where the mutation exists. After this, we can insert a donor template of DNA with the correct edit made, and when the cell repairs the DNA, this edit will be completely edited into your DNA, and that's how we might be able to successfully edit our DNA. So why isn't this being used in clinics right now? Well, like every other new technology, there are some issues. One of the biggest problems with CRISPR is delivery. So efficiently delivering CRISPR-Cas9 to cells can be extremely difficult, especially if you're delivering it directly into your body. It has to be specific, efficient, and we have to make sure that the proteins actually penetrate cell membranes and reach the nucleus of the cell without having off-target effects. So before I dive into the different delivery methods, I just want to explain to you a little bit about what a vector is. So a vector is sort of like a delivery truck. So when we're delivering any type of drug into our body, such as CRISPR, we encapsulate it in a vector, and there's many different types of vectors. So here are some of the main ones being used for CRISPR. So first we have viral delivery, which uses viruses to deliver CRISPR to our cells. So viruses, they naturally have the structures needed to penetrate cells. So basically what we do is we encapsulate DNA and the Cas protein within the virus, and then we deliver it to our cells. Then we have lipid nanoparticles, which, if some of you remember, were used in the COVID-19 mRNA vaccine. And then we have cell-penetrating peptides, which, as the name suggests, are pretty efficient at penetrating cell membranes. So these can be effective too. Now, all of these have their own limitations, but after doing a lot of research, according to my research, I realized that viral delivery may not be as promising as the other methods because it does have some limitations. For example, immune responses. So viral delivery triggers the body's immune system, which can cause a lot of issues. And then, of course, it expresses the CRISPR protein for a bit too long, even after the edit is made. So this can cause a lot of unintended effects. And it's also not very customizable. So the Cas9 protein is, even in size, a pretty big protein. And we also have to deliver RNA along with this. And viral delivery, a lot of times these viruses have limits for cargo size, so this can be a problem. So some companies working in this field include Intellia Therapeutics, and these guys are working with lipid nanoparticles to deliver CRISPR. Then we have Mammoth Biosciences, and they're working with viral vectors, but they're working with smaller Cas proteins such as nanocas to deliver these efficiently inside viral vectors. So what have I been working on? Well, after researching all of this, I decided to apply my learning by creating a visualization of an actual delivery system. So I used some tools like Pymol and AlphaFold to create an actual visualization. And I read this really cool study, and they used lipid nanoparticles in combination with cell-penetrating peptides, and I found this to be really promising. So on the screen, the yellow sphere that you see is a lipid nanoparticle. And within it, the pink protein is the Cas protein, and then the little peptides are the cell-penetrating peptides. So here's another picture of that. So what's next? Well, after meeting with a lot of professionals and after conducting a lot of research, I realized that there probably isn't one method that best fits all applications, because there's so many different types of cells in our body and there's so many different diseases. So even when picking the type of delivery method, we need to take that into consideration to determine the type of peptide to use and the type of system to use. So I learned about this disease called cystic fibrosis, and what I'm going to be working on next is I'm going to be applying all of my learning to cystic fibrosis to see if we can come up with some kind of delivery method to apply gene editing to cystic fibrosis. I'm also really excited to meet with other professionals in the field and maybe even create more visualizations and simulations using online tools. So yeah, that's basically it, and feel free to connect with me on LinkedIn if you would like. But yeah, thank you.